Okay, the stream has started. I'm gonna give just a few minutes since we just started it to allow people to join, and then I'll start the intro.
Sounds good.
That's good.
Okay, we've got a few people joining, so I'm going to get started to make sure that you have all the time that you need to talk, Ron. So hello, everyone, and welcome to the paper trail. My name is Bria Macklin, and I'm a postdoc at the Gladstone Institutes. The paper trail is an online interactive journal club that was developed by 4 former colleagues turned lifelong friends. So Michael Blatchley, who is a postdoctoral fellow at CU Boulder, Franklin Hall, who's going to be your host today, is a graduate student at Johns Hopkins University. Quentin Smith is an assistant professor at UC Irvine. And so, born from our love of discussing science and our yearly paper a day challenge, we came up with the idea for the paper trail to be a platform that allows primary authors to come and share the story of their research. We really wanted the paper trail to stand out from other online presentations. And so to do this, we came up with a set of guiding questions that prompt the author to share more than just the results. And so we're so happy that you've decided to join us for this live stream. And so in true Twitch form, we ask that you use the chat feature to ask questions throughout the presentation. We have moderators, us in the chat, that will relay your questions to Ron. And Ron is eager to answer your questions throughout the presentation. So don't hold them to the end. Ask them as they kind of pop into your head. And so if you do not have access to the chat feature, you can simply create a Twitch login. It creates a few seconds. You can go do it right now and come right back, and that will give you access to the chat feature. And so with that, I would like to introduce our speaker, Ronghui Zhu. And so Ron received his bachelor's in biochemistry from the Hong Kong University of Science, where he did work in structural biology with Professor Mingji Zhang. After finishing his undergraduate study, he went to Caltech and he joined Professor Michael Elowitz's lab as a graduate student. And his thesis work focuses on understanding the design principles of multicellular circuits in mammalian cells through mathematical modeling and experiments. So I'll let Franklin and Ron take it away. And Ron, you can start sharing your slides whenever you're ready.
Perfect. Thank you, Bria. Yeah, we're excited to have Ron with us. And Ron, it's all yours.
Yeah, thank you. Thank you, Bria, for a very kind introduction. And thanks, Frank. So I'm very happy to be here to share our recent work with the paper trail audience and tell you also some of the behind the scenes story about this paper. And so first, I want to thank all the people that directly involved in the project and also provide generous support along the way. So first, my PhD advisor, Michael Elowitz, could not only provide unlimited help and support toward this project, and to be honest, like all the way through my PhD years. And Jesus and Jordi helps with the computational parts of the project. And I want to special shout out to Justin Boyce, who teach the BE150 class with Michael at Caltech. The class name is Design Principle of Biological Circuits. Highly advise you to search that online. I learned most of my modeling skills, and most of the modeling codes are coming from that classes. All right. So in Eloise Lab, we are interested in understanding the design principles of biological circuits, particularly those that programs multicellular behaviors. So for example, during embryonic development, cell growth and death, cell differentiation, self-movement, or signaling are all programmed by these biological circuits. And the circuits I'm particularly interested in are those that control cell-fate decisions. So these cell-fate control circuits control the cells, genetically identical cells, to follow a stereotypic developmental path to differentiate into molecularly and functionally distinct cell types. And the question for us is, could we create a synthetic version of this natural cell-fate control system that have all the nice cell-fate control properties? Okay, so, but you might be wondering, where is this project coming from? And I heard that the paper trial audience loved the project's timeline, so I attached one here. Let's go back to 2017, when I was a second-year PhD student. So at that time, during my whole second-year PhD, I basically just joined Eloise Lab and still continue working on my rotation projects. And at the end of my second-year PhD, I think many PhD students can relate to this, is that you realize that you need to do something that is called candidacy. Essentially, you need to write and present a proposal for your thesis idea and for your thesis project. And this is where the initial multiphase idea and preliminary modeling results comes from. But it is not that straightforward, because at that time, I wasn't that clear about what I want to work on for my thesis. The only thing I know is that I don't want to continue my rotation project as my thesis project. And also, I'm pretty intrigued by how cells make fate decisions. And actually, the paper that attracted me to Michael's lab is this 2013 paper where a former postdoc, Hao Yuan, create and characterize using mathematical modeling and experiments a beautiful cell-fate control circuits that controls blood development. And Michael basically agreed that I should pick a thesis project that I feel truly passionate about. But he is on to slightly different things, essentially how promiscuous protein-protein interactions perform cellular computations. And a related paper that is published at that year is also a former postdoc, Yaron Atepi, describing how promiscuous ligand-receptor interaction can perform complex cellular computations. Okay, so like, as I said, I just realized that I need to do candidacy. So it was a really rush kind of process. I didn't really find a way to align our interests. So basically, for my candidacy proposal, I just follow Michael's idea, proposing engineering a synthetic cellular computation system using promiscuous protein-protein interactions. All right, it is actually a very vague and immature proposal and ideas. And my committee member really grilled me during my committee meeting. But the silver lining is that at the end of my proposal writing, I seem to find a way to align our interests. That is kind of basic, the basic idea of this multi-phase design coming from. So, but this idea is not coming just from nowhere. At that time, I was reading a lot of papers describing the natural cell-fate control system. And here I see two interesting features. For example, the myogenesis circuit on the left here. So first, I found that many fate regulators, so they are often transcription factor or cofactors, dimerize with each other in a variety of different ways. And different dimers can activate or repress different set of genes. And furthermore, some of the dimer can even activate the transcription of themselves, forming a positive autoregulatory loop. So this promiscuous protein-protein interactions and positive autoregulation seems to be two recurring things in many natural self-fate control systems. Another example is the endodermal differentiation system. And based on this, we designed a synthetic circuit called multiphase. And here we have two, and that just extracting out these two prevalent features. Okay, so here we have two transcription factors. We name it A and B. We designed them in the way that they share the same dimerization domain, so they can both homodimerize and heterodimerize. And then we designed the promoter of this transcription factor in the way that they can only be bound by the homodimer, but not the monomer or the heterodimer. So each transcription factor can homodimerize to self-activate. And since the heterodimer cannot bind to either of the promoter, so you just do nothing. So transcription factor can also heterodimerize to mutually inhibit each other's function. Okay, any questions on this part?
Yeah, Ron, quick question. So what is it that actually determines within the cell, what the ratio is going to be of transcription factor A versus B, both being on or one being on or off in vitro or in vivo system?
Yes. So in the multiphase, thank you for the question. It's a really nice question. So for the multiphase in the in vitro kind of in our synthetic circuits, it is determined by a variety of factors. As I will show you just in a minute in the model. So first of all, this promoter by itself, even without the self-activation, can have some basal expression of the transcription factor. And the in vivo counterpart is kind of, if you put it in the in vivo natural circuit context, it is related to, for example, the chromatin state of that gene, whether the chromatin is open or closed or poised positions. Then that will affect the basal transcription of this cassette. And then it is also affected by whether the circuit is self-activated. In the multiphase, much simpler. So you form homodimer, and homodimer can bind to the promoter and activate the transcription. And this will give you a high level of transcription. But in the natural circuit or in the natural system, it's more complicated. It can be affected by a variety of different factors. So first of all, you not necessarily see this autoregulation. So just one gene activates itself. It could be a more complicated network. For example, like A activate A plant. So it's another gene and activate another gene, and that gene goes back to activate A. So it could longer or bigger positive feedback loop. And also the natural protein is also subjected to multiple level of modification. So for example, this in the natural system, this dimerization process could be modulated by protein phosphorylation, for example, and also the mRNA can be subjected to microRNA kind of regulation. So in the natural circuits, it could be more complicated, but in the multiphase, we're trying to extracting out some kind of simple feature out of these complicated natural circuits.
Awesome. Yeah, thank you. Great response.
Thank you. So what we like about this circuit design is that it's simple enough that we can build a tractable mathematical model to describe its behavior. And here we can use one set of equations to describe the protein dimerization process and another set of ordinary differential equations to describe the positive autoregulation and protein removal. And analysis of this model gives rise to a variety of different multistability regimes. So for example, here I'm showing one interesting tri-stable regime where you have 3 stable cellular states or 3 attractors shown by these 3 filled black dots on this phase diagram with transcription factor concentration as axis. And the shaded regions is the attractor basin. And so any cell in a certain basin will be attracted to its corresponding stable cellular states. So here I want to give you some intuition on what's happening here, why there are 3 stable cellular states. So these 3 states differ by their transcription factor expression and as a result, their dimer composition. So for example, on this bottom right state, we call it A-only state. You will have a lot of A expression, so they can form a lot of AA homodimer and self-activate. And any basal B expression will be quickly neutralized by forming heterodimer with A, so it cannot self-activate. And similar thing happens in this B-only state. And in this middle A plus B state, both A and B are expressed at high level, so they can form a lot of heterodimer, but also enough homodimer to each self-activate and sustain their high expression. So this shows that, at least in the model, the multiphase can generate multiple stable cellular states. So this is the first multistability property we're aiming for. Any questions?
Yeah, so one quick question would be, are these fates transient, or are they pretty set? Once you reach a plateau or a level of stability, do you still see kind of movement back and forth, or is it pretty stable?
Yes, so this is a great question. So that depends on kind of, so if you're asking in the model, though that depends on whether you are using a deterministic model or stochastic model. Okay, so for a deterministic model, once the trajectory reach this steady state, the definition of steady state is that once you reach it, you won't go out unless you perturb it with some very strong perturbations. But if you use a stochastic modeling that counting the biological noise, and that's also more kind of more realistic modeling methods that reflect what really happens in the in vivo system, that you can have some noise based on, for example, transcriptional noise, or even protein-protein dimerization noise or degradation noise, then you will see the cells jiggering around in different states. And if this noise is large enough, you could see some spontaneous transition from one state to another state, even without kind of external perturbation. But if you perturb them in the natural system, as I will show you later, you can switch the cell from one state to another state. Yeah.
Okay, awesome. Maybe if we have time at the end, you could talk a little bit about how you kind of implement the stochasticity into your coding, your methodology.
Yes. Cool. So so this is just a modeling, and modeling shows that it works. So next natural question is, how can we implement this experimentally? So ideally, we will characterize different synthetic components to see which one works, which one doesn't work, and to see which one we can use to build the multiphase. But the reality is, I was still working on my rotation project. So I really don't have that much time to test different multiphase components in the background of my rotation project. And the mentality at that time was, for my rotation project, if I can finish it as quick as possible, then I can publish a quick paper. Then I can start working on my thesis project. So this is kind of a guarantee paper. Then you can work on more risky projects. But after 1 year, both Michael and I found that it is really not sustainable, because as much as we want to finish this rotation project as quick as possible, we could not, because we are hitting some technical difficulties and some bottlenecks that we really cannot break through at that time in a short time frame. So we have to make a very hard decision to pause the rotation project and fully commit it to multiphase. In retrospect, this is really the right decision to make, but like at that time, this is really a hard decision, because you kind of relinquish some projects that you already made some progress on to jump into another project that has a lot of unknown components. But after we fully committed to multiphase, we did make considerable progress with some minor setbacks, which I'm going to show you later. So to make multiphase experimentally, we need transcription factors that can self-activate only as a homodimer and inhibit each other through heterodimerization. and ideally could allow us to externally control their functions and abundance. And long story short, we choose zinc finger DNA binding domain as our basic building block. There has been decades of work on these zinc finger DNA binding domains, and people have engineered synthetic zinc finger arrays that consist of 3 zinc finger that can recognize a 9 base pair DNA binding regions. And importantly, in mammalian cells, studies from Pamela Silver's labs and others shown that if you fuse this zinc finger DNA binding domain with an transcriptional activation domain, as a monomer, it does not have enough binding affinity to bind to this 9 base pair region to activate the transcription. But if you incorporate a dimerization domain, in this case, the GCN4 dimerization domain, it can induce the dimerization of the transcription factor, and this dimer can recognize a longer 18 base pair DNA binding regions with sufficient binding affinity to strongly activate the transcription. So we test this in our hands using a fluorescent reporter, and here you can see that, as expected, the dimer strongly activates the transcription while the monomer barely activated. And we further introduced arginine to alanine mutation to completely eliminate the monomeric activation while still preserving much of the dimer activation. So this shows that these engineered zinc finger transcription factor can activate the transcription in a dimer-dependent manner. So next, we want to see whether this transcription factor can be controlled to self-activate. So to add controllability, we switch out the GCN4 dimerization domain with an FKBP inducible dimerization domain so that we can use the drug AP1903 to control the dimerization. And at the C-terminus, we fuse an DHFR diagram so that we can control the protein degradation by another drug, trimethoprim. And to test the self-activation, we simply design a simple self-activation circuit with a co-expressed fluorescent protein for readout. Okay, and as expected, without AP1903 to dimerize the transcription factor, the circuit cannot self-activate. And with AP1903, the circuit can self-activate, and the self-activation strength can be controlled by the concentration of trimethoprim. So this shows that these new engineered zinc finger transcription factor can self-activate in a dimer-dependent and controllable manner. Okay, any question on this part?
How hard was it to select the zinc fingers that were either going to be homodimers or would inhibit each other as heterodimers?
Yes, so essentially, all our tested zinc finger works in the 2 paper that shows here. So the only difference is that for different zinc finger, we need to introduce different numbers of mutation to realize this kind of ideal effect. So you can see for this zinc finger, the monomer has some but weak activity. And when we introduce 2 mutation, it completely eliminates this monomer activation.
Oh, okay.
So everything is dimer dependent. But for some other zinc finger, we have to introduce either 1, 2, 3, or even 4 mutations to these 4 arginine sites to make them into alanine to completely eliminate their monomeric activation. So basically, all the zinc finger works. It's just how many mutations you need to kind of introduce. And we did a small batch screen on that.
Okay, cool,
yeah.
Awesome.
Yeah. And the final criteria is that this zinc finger transcription factor can inhibit each other through heterodimerization. To test this, we introduced a second transcription factor with a different zinc finger DNA binding domain into a self-activation cells. And you can see here, only the second zinc finger transcription factor with a matching dimerization domain, in this case, the GCN4 dimerization domain, can significantly inhibit the self-activation, but not one with a mismatch or no dimerization domain. So this shows that this transcription factor can inhibit each other through heterodimerization. Okay, so with this transcription factor matches with all our criteria, we are ready to build the simplest multiphase circuit with 2 transcription factor self-activation cassettes. And the differences between these two cassettes is that first, they use two fluorescent protein for readout. And second, they use two different zinc finger DNA binding domain and corresponding DNA binding site. And to make the multiphase cell line, we stably integrate these two cassettes into a mammalian cells and select monoclonal cells for characterization. Okay. So before I show you the positive result, I want to show you some small kind of detour we ran into that take us a few months to figure out. So to make multiphase that realize this interesting tri-stable behavior, we need transcription factor that behave relatively similarly. So in the model, we model them to be behaving exactly the same. But in real life, no transcription factor will behaving exactly the same. So we just choose two that behave like reasonably similar with each other. But when I say choose two, we actually don't have a choice because we only characterize two at that time. So we only have like two zinc finger at our disposal. And from preliminary assay, we found that these two zinc finger behave kind of similarly. So we just proceed with these two zinc finger. But when we built the cell line, it seemed the cell line just behaved really weirdly. And then we go back to the assay that characterized these two zinc finger and found that the assay we're using to test these two zinc finger is not sufficient. So what assay were we doing at that time? So we try to quantify the transcription factor self-activation strength by this simple assay. So here I show you the self-activation circuit before. So we stably integrate this self-activation circuit into the mammalian cells and make a polyclonal cells for characterization. So here, without the drug to dimerize the transcription factor and stabilizing it, you can see that the cells have very low level of citrine. And when you add these two drugs to permit self-activation, part of the population will become citrine positive, suggesting that part of the population can self-activate and strongly activate this cassette. So we use this percentage of citrine positive population as our quantification for the self-activation strength. But later we realized that this high citrine percentage could mean a combination of two things. And depending on different zinc finger, these two things can be very different. So if you have a very high citrine positive percentage, it could mean that you have a very strong self-activation. This means that the self-activation strength is very strong, so you don't need that much transcription factor to start with to start this self-activation loop. But another possibility is that you could have very high basal expression level even before the self-activation. So this means that even without any transcription factor binding to the promoter, this promoter could have very high basal expression level that could be introduced by this zinc finger binding site. And then since you have more starting basal kind of transcription factor to work with, then even if you only have a weak self-activation strength, it can start this self-activation and resulting in a similar citrine positive population. But at that time, we didn't consider this possibility because all the zinc finger binding site, we kind of cross-reference that into the CHO genome. There's no kind of this kind of binding site in the genome. So it's kind of wild to think about, any random binding site you add to the promoter will change its basal expression level. But, biology is that weird. So it seems that there are some questions from the audience.
Yeah, a couple of questions just about your selection of the cell line you use. So one question is, are the CHOK1 cells, they're a hamster epithelial line? Was this tried in any human lines?
Yes. So right now we are kind of people in the lab picking up this project is testing that in various other cell lines. Yeah. But in this particular project, we only test the CHO line. Yeah.
Was there a particular reason you chose the Cho line? Just curious.
So one particular reason is just for realistic reason, practical reason. Those Cho line. So for the multiphase, to kind of when we characterize the multiphase, we really want the monoclonal lines. So because we want to see the cells with genetically identical, phenotype, like with the identical genotype will exist in different, stable regions. So we want monoclonal line that requires the cell to grow from, single cell into a colony. And that's what we did in the experiment. And the Cho line has been known to, grow very fast starting from single cell into a colony compared with many other lines. So even fast, like kind of growing cell line like hack cells, they don't like to be alone and in a single well to grow from single cell into a colony. But the Cho line is really fast. So that allowed us to have a fast iteration of the project. Yeah.
Oh, okay. Awesome. Thank you.
Yeah. Cool. So so biology is that weird. We found that two zinc finger we use actually behave exactly the opposite in terms of these two metrics. One of the zinc finger have a very strong self-activation, but low basal expression of the promoter. But the other zinc finger have very weak self-activation, but its binding site actually introduces additional basal expression to the promoter, which is a big surprise to us. And it's a bug, but we turn that into a control handle in the future experiment. Because in the future experiment, we are not using this zinc finger DNA binding domain in our cassette, but we still use its binding site, this GAC, GCT, GCT 9 base pair binding site as some cryptic DNA sequence that we can add to the promoter to modulate the basal expression of that promoter. And this basal expression is actually an important parameter alpha in the model that actually previously we don't have a way to tune it. But since we found that this binding site actually introduce additional basal expression to the promoter, we just use that to modulate the basal expression of our promoter. Okay. So so this is kind of a small detour, but like in the next week, trying to characterize a new set of zinc fingers. So here I want to thank Mo Khalils for his suggestions on the zinc finger that I can use from his 2012 paper. And also we also characterize a few zinc finger from another paper from Pamela Silver's lab. And once we get this new set of zinc finger, we use a more rigorous assay to characterize each of them and found the two zinc finger transcription factor that behave truly similarly with each other. And just everything just began to work. And within 3 to 4 months, we already got our first tri-stable multiphase 2 lines. And this is one of the key results in this paper. So here I want to show you, what this like multiphase 2 line looks like. So again, we have this stably integrate these two cassettes into the mammalian cells and select monoclonal cells for characterization. And since these two cassettes have two different fluorescent proteins, we can use flow cytometry to characterize them. So here, x-axis is the mTOR representing A expression, and y-axis is the citrine representing B expression. When we add a drug to permit dimerization and stabilizing the protein, the cells can spontaneously go to 3 distinct clusters, differ by the expression of the two cassettes. And this is really encouraging because as you can see here, these 3 distinct clusters already looks very similar to the tri-stable regime predicted by the model. And to see whether the cells are stable in these 3 different states, we use cell sorting to separate them in 3 different wells and continuously culture them in the drug for extended periods of time. And here you can see that the cells are stable, extremely stable in all these 3 states within this 18-day periods, as shown by majority of the cells remain in their originally sorted state. And we can directly visualize their stability by using time-lapse microscopy, imaging them growing from single cell into colony in 3 different states, showing by 3 different fluorescent color combinations. So here you can see that since majority of the cell remain in their originally sorted states as they grow and divide, they form this very nice patch-like pattern at the end of the movie. So this shows that both in the model and in the experiment, the multiphase can generate multiple stable cellular states. So this is the first multi-stability property we're aiming for. Okay.
I have to say, man, that's really cool video, cool image as well. I'd love to have that as a background.
I have a cooler video later. Hold on. Yeah.
All right.
Yeah. Cool. So so I think for many people that doing projects and you get a key results, this is sometimes that this project will lead to somewhere. You can already see the light at the end of the long tunnel. So this is kind of so at that time I already know that I'm onto something. And the next is just I need some time to demonstrate different properties of the multiphase and do different control experiments. Okay, so during this 1 year of time, we demonstrate a few more properties of the multiphase. One of the property of the multiphase that the multiphase is expandable. So one unique feature of the multiphase that we can it implement this mutual inhibition at the protein level through heterodimerization, which makes the circuit easily expandable simply by adding a new transcription factor self-activation cassette. Since the new transcription factor will share the same dimerization domain with the existing transcription factor, it will automatically join this mutual inhibition at the protein level. So this means that we can expand the circuit without re-engineering the DNA sequence of the existing cassette. And this is in contrast to imagine an all-transcriptional design. So here you have 2 transcription factors mutually inhibit each other at the transcriptional level. If you want to expand the circuit, you need to re-engineer the DNA sequence of the existing cassette so that this new transcription factor can bind to. So we first test this expandability idea in the model. So here you can see that in the model, showing this expanded multiphase circuit with 3 transcription factor, it can generate as many as 7 stable states. Any questions?
Yes. Yeah, so was the ability to expand this so easy? Was that something that you guys pre-programmed, or was it like a byproduct? You guys just tried it out and it was just very easy?
Yeah. So this is actually so again, the multiphase, we didn't try it out until more than a year after the modeling. So this is really something I would say not initially designed, but also not until we try it out we found it. So it's kind of in between. So initially we didn't think about this, but when we begin fully committed to multiphase, testing different components, and at the same time, I'm trying to enrich this modeling part of the project. So when I do more modeling part, I found that this model can be easily expanded. And then I look at the design, I found that this design can also be easily expanded. So this is kind of interesting kind of correspondence between this model expansion and the design expansion.
Okay, yeah, awesome.
Yeah. Okay, so to experimentally implement this idea is also pretty simple. We simply integrate a 3rd transcription factor self-activation cassette into an existing multiphase 2 line to make a monoclonal multiphase 3 lines. And to directly visualize the stability of these multiphase 3 lines in 7 different states, shown by 7 different fluorescent color combination, we use time-lapse microscopy again to directly visualize the cells growing from single cell into a colony. And since, again, majority of the cell remain in their originally sorted states as they grow and divide, they form this very nice patch-like pattern of 7 different colors at the end of the movie. So this shows that the multiphase can also being easily expandable to include more transcription factor and generate more states. Okay, so this is one additional property we are aiming for. The other property we are aiming for is the controllability. So the natural selfie control system can not only establish multiple stable cellular states, but can also control the cell to traverse through these states. And we want to see whether multiphase can do similar things and recapitulate some of the interesting phenomena we see in the natural selfie control system. For example, many cell differentiation process is largely irreversible. So when we analyze the multiphase model, we found that it can operate in different multistability regimes. So on the left is the tri-stability regime I just showed you. When we reduce the protein stability, and we can do so in the experiment by reducing the concentration of trimethoprim we add to the medium, we can specifically destabilize this middle A plus B states, and we are left with a bistable regime with only A only state and B only state. And here I want to give you some intuition on what's happening here. So as I mentioned before, these three states differ by their dimer composition. In particular, these middle states have a lot of heterodimer formation, and as well as just enough homodimer formation to each self-activate. But when you reduce the protein stability, the global protein amount also reduces. So this means that this middle state can no longer form enough homodimer to each self-activate. So the cells have to make a choice to go to A only or B only state, where since there's no competition to form heterodimer, they can still form enough homodimer to self-activate. Okay, and then we use stochastic modeling to confirm our intuition. So here we model the cells as these red dots starting from this A plus B states. And here when we reduce the protein stability, they spontaneously escape from the destabilized A plus B state to go to A only or B only states. And interestingly, this process is irreversible, because even when we put the cell back to the high TMP conditions, the cells are trapped in this A only and B only state and cannot go back to this restabilized A plus B state. And if you think about this A plus B state to be the pluripotent state, and this A only or B only state to be the differentiated states, these behaviors recapitulates what happens when you transiently withdraw the pluripotent condition from the pluripotent stem cells, and this will lead to irreversible cell state transitions. And we can also test this in the model. We can also test this in the experiment by putting the sorted A plus B cell from the high TMP condition to the low TMP condition. And here you can see that the cells spontaneously escape from the A plus B state to go to A only or B only state and cannot go back even when we put them back to the high TMP conditions. So this shows that we can, in multiphase, we can modulate the stability of individual multiphase states and recapitulate these irreversible cell state transitions. Okay, any questions?
Yeah, one question is, I know in the paper you discussed removing AP1903 in TMP to kind of reset the cells and reset the kind of system, right? Yeah. And then you can get it back to tri-stability or bi-stability from there. Mm-hmm. In this case, though, by just lowering and raising the TMP, you can create irreversibility. What's the difference between those two processes?
Oh, so it is really kind of, yeah, this is a really good question. So this is kind of different parameter regime kind of tuning. So here, when you think about if you remove both the AP1903 and TMP, what will happen is that everything will be, all the states will be destroyed, and you are only left with a monostable regime where only, just low expression of both transcription factor is stable. And in that case, when we the cells starting from here, right? And then when we add AP1903 and TMP back to the system, since around here, there's multiple routes that you can go to A only, B only, or A plus B state, you could still go to all three states. But what happens in this case is that when you gradually reduce the TMP, all right, and then the cells, goes to the A only and B only states. And then when you put the TMP back, you can see that these cells are trapped in this kind of attractor basin. And when this attractor basin evolve, they are trapped in this basin and cannot just jump to this blue basin. So because this basin kind of create a boundary between them. So this results in the kind of the cells trapped in A only and B only state. But imagine if you, for example, if you reduce the TMP, all right, but you don't wait, okay, you put the cell back here, then imagine like you put the cell back here, and then these blue regions reappear, right? And the cells haven't leave the kind of the territory of this blue region, then it is possible that the cells can go back to this A plus B state. So it is really the modulation of this attractor basin that create this kind of irreversible cell state transitions. Yeah. Any other questions? Okay, cool. So yeah, so this is kind of basically during this whole years the two additional property we are aiming for. But I think people may already notice this between these two time points, we also all witness another thing that happens in the world, that is the global pandemic. And this multiphase project is also not immune to the pandemic. And I would like to share some just some side story about how pandemic affected this multiphase project. So overall, the project is not largely disrupted, but it still introduced some inconvenience. And what makes the things more complicated is that our lab also moves from one building to another building. And here I want to shout out big thank you to my lab mate and my friends, Felix and Xun, that helped me, through this process. For example, for Felix, at that time, towards the end of the multiphase experiment, I was in contact with a person that tested positive. So I have to quarantine for a week in my home. But at that time, I was doing this long-term measurement of the stability of multiphase states that lasted, we're aiming for more than 30 days, 30 to 36 days. And the experiment already went on for more than 20 days. So we don't want to restart the experiment. So I have to reach out to Felix and ask him to help me to measure a few time points. So these cells have to be measured by flow cytometry every 3 days and continuously passage them in different kind of drug conditions. So I draw out, and another thing is that at that time we are isolated in different tissue culture room just for, prevent COVID spread. So the tissue culture room actually I'm working on, he is not very familiar with. So I have to draw out this kind of room layout as well as my plate laid out to him to tell him, where everything is, how to, measure, how to do the experiment. So I really appreciate this kind of memories that coming out of this. Okay, so after this, after we finished the experiment, it took us about 3 months to write out the manuscript, and Michael and I kind of wrote side by side virtually with each other on that. And we submit the paper to the journal and got review back after 1 month. And then it takes us another 5 months to do the revision, and then we resubmit, and the paper was accepted after another 2 months. Okay. Any questions on this part?
Yeah, we actually had a couple of questions from the chat I wanted to go through. One was, could multiphase be coupled to a secretion system where the state space would be modulated by the secreted signals?
Yes, and this will be touched on in the final slides of my presentation. Yeah, this is kind of our future direction we're thinking about.
Okay. And then the second question is, if TMP forms a gradient, would cells form a tissue-like structure just like BMP gradient during embryo development?
Yes. Okay, so this is super interesting question because I'm less confident about TMP forming gradient because the TMP is a small molecule, so it just diffuse I would say too fast. So I wouldn't say it will form gradients per se, but the BMP could form gradient, for example, and all other signaling molecule could form, form gradient. And I didn't say what my rotation project is. My rotation project is about studying this, morphogen gradient, and it's about BMP gradients. And if we can connect the BMP gradients with the multiphase, for example, use BMP to control multiphase, I think we could see some really interesting, morphogenesis, synthetic morphogenesis like phenomenon. So these are great questions.
Yeah, thank you to the chat for the questions. Please keep them coming.
Yeah, thank you. Okay, so I think audience may also curious about the review process. So luckily, most of our reviewers generally like our paper, and most of the reviewers' comments are reasonable or at least addressable, and we'll address most of them. So just one example, in the first draft, we're trying to show that we can control the switching of the cells from one state to another state by using different chemical inducers, for example, like doxycycline or tamoxifen, but only in the bistable regime between two states. And the authors asked why not also showing that in the tristable regime. So that's what we did in our revisions, and in our final draft, we shall also add this switchability in the tristable regime. And I think this really kind of strengthened our switchability arguments. But not all the reviewers' comments are addressable. So some unaddressable comments, I think, originated from reviewers not realizing the limitation of multiphase. And what we did is that we explained the limitation and trying to meet them in the middle. So for example, one example is that in the model, we only model the symmetric case, meaning that we assume all the transcription factors to behave exactly the same. But as I mentioned before, as much as we want to make them exactly the same or as similar as possible, they still have some discrepancy between the behavior of the transcription factor. This will result in asymmetry behaviors of the system. So for example, for this cell line, when we add AP1903 and TMP, the cells goes to A only state and B only state. But by the model, it should go to these two states with a 50-50 chance. But in reality, you can see that it favors A only state over the B only state. And the reviewer asks whether we can predict what cell fractions the cells will go to during this kind of perturbations. And this is not what we can do in the multiphase model because we didn't measure each multiphase parameter experimentally in our particular experimental systems. And we only use a physiologically reasonable estimated parameter from previous literature. So we only have a range of order of magnitude of each parameters. So the current multiphase model, we can only do qualitative prediction. For example, we know the current multiphase design can realize tristability, it can realize bistability, and we can transition between these two multistability regimes by modulating the protein stability. But we cannot make quantitative prediction, such as how much protein stability we need to modulate to transition between these two regimes, or what the reviewer asked, what are the resulting cell fractions when the cells exit from an unstable state. So we cannot predict all experimental results, but we could explain our experimental result in retrospect by modifying the multiphase model to incorporate this asymmetry between transcription factors. So the major change between the first draft and the final draft is that we incorporate these asymmetry parameters into our multiphase model. to make this asymmetry multiphase model and use this model to fit the experimental data. And you can see that with this asymmetry parameter, we're doing a pretty good job of fitting between computational result and experimental results. So here we at least partially address reverse comments. Okay, so just as a summary, today I introduced the multiphase, and this circuit design really inspired by the natural features, the features from natural self-weight control systems, namely the promiscuous protein-protein interaction and the positive autoregulation. It can establish multiple stable cellular states, and we can control the stability of individual states and recapitulate these irreversible cell state transitions seen in many natural differentiation process. It allowed us to expand the circuit to support more cellular states. And remarkably, all the experimental results matches very nicely with our model predictions. And we think this multiphase can provide a basis for future synthetic multicellular systems. And this is related, and going forward, this is related to what some people was asking about using some signaling pathway to control multiphase. This is exactly what we're thinking. So in the first paper, we demonstrate that we can establish and control the cell to switch between state using chemical inducers, such as doxycycline or tamoxifen. Going forward, there are many natural or synthetic cell-cell communication systems at our disposal that we could use to connect that with multiphase. We want to see whether we can use this cell-cell communication system to control the differentiation of multiphase cells. And once the cells go to different cellular states, they can express different signaling modules and allow them to communicate with each other and coordinate for some therapeutic functions. And I'm excited that in the lab, there are already several people picking up different aspects of this project. Okay, so any questions?
Yeah, I mean, this was really interesting, great talk. We just have a couple other questions. And one was, when you think about when we talk about the review process, you posted this on bioarchives. And so how did you decide when to post a paper there? And did you get any feedback after you posted the paper there?
Oh, yeah. So yeah, this is a great point. We didn't sort of decide whether to put on bioarchive. I think it's almost natural to most of people now, unless there are some, I don't know, preparatory concern, but we don't have that kind of concern. So we just, posted, and it's bioarchive made it very easy to, just put it there. So it's kind of very convenient to do. And after that, we see a lot of feedbacks, and particularly one interesting trend of the feedback is that in the paper, in the first paper, we mainly show, how stable each state is. We do this long-term experiment showing that, once the cell is in a certain state, it remains stable for, however long time. But increasingly, I found that people are actually maybe a little bit more interested in, how can you switch the cells, transition the cell between states? And this, more and more people asking this kind of question that also kind of guide us to think about our future step of the multiphase. How can we design this control system to control the cell between different states? So this is kind of the main things.
All right, awesome. Do we have any other questions from the chat?
Yeah, as we are waiting for the questions, I can show the final slides, which is something I particularly like about this story, is that I said before that we have to make a hard decision to pause the rotation project and fully committed to multiphase. And once this multiphase is finished, also like at least from my side, I was able to circle back to my rotation projects. And recently, we just got some exciting results from that project. And hopefully we can finish that this time really pretty soon. And can't wait to sharing these new results with you.
Oh, wow. Awesome. Congratulations, man. That's exciting.
Yes, this is exciting.
All right, well, we're coming up on the hour. Hopefully you have some questions to chat. If we don't, we really want to thank Ron for being here today. Bria's going to drop his Twitter so you can see any of his new stuff that's coming out, hopefully in the near future. Please give him a follow there. And if you have not followed or like to subscribe to the Paper Trail on Twitch, please do so. We definitely plan to increase the amount of events that we hold. And Ron, if you don't have anything else, Bria, if you want to take us out.
Yeah, thank you so much, Ron. That was such a great talk. We're going to end the stream. And as Franklin said, I'll put Ron's information in the Twitch chat. So if you want to stay on just after the stream, it'll be in there. Yeah, so I'm going to go ahead and end this stream. Thank you for joining.